Alrighty, this presentation is in response to exam 1C, question number 5 concerning the structure of antibodies and their types. So to start off, I'll point out that an antibody is it's a protein produced by the immune system, specifically produced by plasma cells. Their job is to neutralize pathogens such as bacteria and viruses. Concerning their structure, immunoglobulin or antibodies are large, by large, in the neighborhood of 150,000 Daltons, macromolecules. They're globular plasma proteins. They have a very distinctive Y shape, and we'll get into that in just a second here. They have sugar chains added to conserved amino acid residues. That is to say, antibodies are glycoproteins. The attached glycans are critically important to the structure and function of the antibody. Among other things, the expressed glycans can modulate an antibody's affinity for its corresponding FCRs. The basic functional unit of each antibody is a monomer, an immunoglobulin monomer, although secreted antibodies can also be dimers, tetramers, or even pentamers. So the monomer is a Y-shaped molecule that consists of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains, connected by disulfide bonds. Each chain is composed of structural domains called immunoglobulin domains. These domains contain about 70 to 110 amino acids and are classified into different categories according to their size and function. They have characteristic immunoglobulin fold in which 2 beta sheets create a sandwich shape held together by interactions between conserved cytosines and other charged amino acids. There are 5 types of the heavy chains. These are denoted as alpha, beta, epsilon, gamma, and delta. There are 5 types of heavy chains, denoted by alpha, delta, epsilon, gamma, and mu. The type of heavy chain present defines what type of antibody it is. So you have A, D, E, G, and M antibodies, alpha, delta, epsilon, gamma, and mu. Distinct heavy chains differ in size and composition. Alpha and gamma contain about 450 amino acids, whereas mu and epsilon have about 550. Each heavy chain has 2 regions, one constant and one variable. The constant region is identical in all antibodies of the same type, but differs across types. Heavy chains gamma, alpha, and delta have a constant region composed of 3 domains and a hinge region for flexibility. Heavy chains mu and epsilon have a constant region composed of 4 immunoglobulin domains. The variable region of the heavy chain differs in antibodies produced by different B cells, but it's the same for all antibodies produced by a single B cell or B cell clone. The variable region of each heavy chain is approximately 110 amino acids long. So in mammals, there's 2 types of immunoglobulin light chain, which are called lambda and kappa. Remember, there's 2 identical light chains in each immunoglobulin molecule. A light chain has 2 successive domains, one constant, one variable. The approximate length of a light chain is about 211 to 217 amino acids. Each antibody contains 2 light chains that are always identical, only one type, kappa or lambda, are present in each antibody. The other types of light chains are found in other vertebrates, but not in mammals. So, some parts of an antibody have the same functions. The arms of the Y shape, for example, contain the sites that can bind to antigens, and therefore they can recognize specific foreign objects. The region of the antibody is called the fragment antigen binding, or Fab region. It is composed of one constant, one variable domain from each heavy and light chain of the antibody. The paratrope is shaped at the amino terminal end of the antibody monomer by the variable domains from the heavy and light chains. The variable domain is also referred to as the FV region and is it is the most important region for binding to antigens. To be specific, variable loops of beta strands, 3 each on the light and heavy chains, are responsible for binding to the antigen. These loops are referred to as the complementarity determining regions, CDRs. The structures of these CDRs have been clustered and classified. In the framework of the immune network theory, CDRs are also called idiotypes. According to immune network theory, the adaptive immune system is regulated by interactions between those idiotypes. The base of the Y shape plays a role in modulating immune cell activity. This region is called the fragment crystallizable region, or FC region, and is composed of 2 heavy chains that contribute 2 or 3 constant domains depending on the class of the antibody. Thus, the FC region ensures that each antibody generates an appropriate immune response for a given antigen by binding to a specific class of FC receptors. By doing this, it mediates different physiological effects, including recognition of opsonized particles, lysis of cells, and degranulation of mast cells, basophils, and eosinophils. In summary, the Fab region of the antibody determines antigen specificity, while the FC region of the antibody determines the antibody's class effect. Since only the constant domains of the heavy chains make up the FC region of an antibody, the classes of heavy chain in antibodies determine their class effects. Possible classes of heavy chains in antibodies include alpha, gamma, delta, epsilon, and mu, and they define the antibody's isotype, respectively. This infers different isotypes of antibodies have different class effects due to their different FC regions binding and activating different types of receptors. Possible class effects of antibodies include opsonization, agglutination, hemolysis, complement activation, mast cell degranulation, and neutralization, though this class effect may be mediated by the Fab region rather than the FC region. It also implies that Fab-mediated effects are directed at microbes or toxins, whilst FC-mediated effects are directed at effector cells or effector molecules. So we can see that the structure of immunoglobulin antibody is directly related to the functions of that specific antibody.